Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Hello everyone. In this presentation, we are going to explore the concept of molecular dynamic simulation of quercetin using Chimera. To start with, let me introduce our team members, which consist of me, Azlia, Farahannisa Zuhdy, and Laily Salsabila. So, what is molecular dynamics? Molecular dynamics is a computer simulation technique involving the generation of atomic trajectories of a system using numerical integration of Newton's equation of motion for a specific interatomic potential defined by an initial condition and boundary condition. The simulation of the motion is realized by the numerical solution of the classical Newtonian dynamic equations. The set of possible atom locations gives, for example, conformational ensemble profile for a given molecule. Molecular dynamics can also provide information on the thermodynamic and dynamic properties of the molecules. The molecular dynamics also can be used for simulation of protein shapes and refinement of X-ray structures. So let us firstly talk about the force field, which is the heart of the molecular dynamic simulation. The forces in a molecular dynamic simulation are calculated using a model known as a molecular mechanics force field, which is fit to the result of quantum mechanical calculations and typically to certain experimental measurements. Force field can be understood as an empirical set of energy functions that helps us get an understanding of the energy related to the interaction between atoms. Typically, a force field is the summation of bond and non-bond terms or covalent and non-covalent interaction among the atoms and molecules as shown in the equation. So the first term is the bond stretching or E stretch and angle bending or E bend. The energy near equilibrium can be approximated by using harmonic potential, which doesn't allow the breaking of bonds. This force constant can be determined by vibration spectroscopic studies. The next one is the torsional term or E torsion, which originates through space and accounts for the rotation of covalent bonds. This approximation of this term can be done with the help of a series of geometric functions. Next is the electrostatic terms or E electrostatic. It is calculated using Coulomb's law with the inclusion of partial charge which are calculated by quantum mechanics. And then we have van der Waals term, which is E van der Waals. It describes the interactive and repulsive interactions between atoms, or in simple terms, the interatomic forces. This term can be approximated by using Lennard-Jones 12-6 potential, which can be thought of as a function of the distance between the centers of the two interacting atoms or the molecules. And the last is the hydrogen bond term, or E hydrogen bond, which describes the energy between atoms that have the potential to form hydrogen bonds. It is approximated by using 12-6 potential, which is similar to the Lennard-Jones potential, but the attractive interaction between atoms disappears faster in this case. In the numerical analysis, Verlet algorithm can be used to integrate the Newton's equations of motions. It describes the two third-order Taylor series expansions for the position of molecule RT, one forward and another backward in time without using explicit velocities. The advantage of using Verlet algorithm is first, it is straightforward and self-starting, and the new position can easily be obtained from the current and previous positions. And second, it requires less computer memory. However, Due to the lack of an explicit velocity term, it is difficult to obtain the velocity at the current positions until the position has been computed for the next step. Another type of numerical analysis is leapfrog algorithm, which was developed to control the error obtained due to the use of velocity varlet algorithm. In this algorithm, positions and velocities are calculated at interleaved time intervals. The advantage of this algorithm is that the velocities are explicitly calculated. However, it is not calculated at the same time as the positions, because of which it is difficult to calculate the total energy at any time point. The next is the molecular dynamics softwares. The first one is AMBER, which stands for Assist Model Building with Energy Refinement. It was developed for refining NMR structures. The name AMBER refers to both force field for the simulation of biomolecules and the molecular dynamic program package. The most widely used force field versions are AMBER 94, AMBER 99SB, and AMBER 03. The second one is CHARM, which stands for Chemistry at Harvard Macromolecular Mechanics. It is also refers to both force field and the program package. It was originally developed by Martin Karplus of Harvard University, USA, and it is the oldest biomolecular molecular dynamic simulations. Provides web-based graphical tools for setting up input files for the simulations. Next, we have GROMACS, which stands for Groningen Machine for Chemical Simulations. It is typically runs 3 to 10 times faster than other molecular dynamics programs. Unlike AMBER and CHARMS, GROMACS doesn't have its own force field. Instead, it can import AMBER, CHARM, GROMOS, and the OPLS force field to run the MD simulation. And then last, we have NAMD, which stands for Nanoscale Molecular Dynamics. It is developed by the Theoretical and Computational Biophysics Group at the University of Illinois, Urbana-Champaign, USA. It is designed to efficiently run on parallel machines for simulating large molecules. The program has high compatibility with other molecular dynamics programs, such as CHARM, since NAMD has the same input, output, and force field formats as CHARM. Let us move on to the application of molecular dynamic simulation. So, molecular dynamic simulation can determine the structures and movement of biomolecules. It also gives assessment of accuracy and refinement of modeled structures, and also for its flexibility of molecules. Molecular dynamic simulation can also used to model the drug receptor interaction and also the protein foldings. Last, the molecular dynamic simulation can also used to understand how mutations affect interactions. And here are the advantages of using molecular dynamic simulations, as no assumptions are made about the process or mechanism to be investigated, and can treat both ligand and protein in flexible way, consider the solvation effect, and predict the accurate binding free energy. And third, with reasonable hydrogens, Verlet's algorithm conserves energy. In this particular presentation, we use quercetin as our main compound. Quercetin is a plant pigment and is one of the important bioflavonoids, especially called a flavonol, present in many plants and foods, such as red wine, onions, green tea, apples, and berries. Quercetin is a bioactive compound that possesses many biological mechanisms. Quercetin possesses an antioxidant and anti-inflammatory effects that might help reduce swelling, kill cancer cells, control blood sugars, and help prevent heart disease. For its application, quercetin is most commonly used for conditions of the heart and blood vessels and to prevent cancers. It also has many applications in terms of its medicinal use. The name quercetin comes from the Latin word quercetum, which means oak forest, which belongs to the class called flavonols that cannot be produced in the human body. And quercetin is yellow in its color and is poorly soluble in hot water, quite soluble in alcohol and lipids, and is insoluble in cold water. To study the structure of quercetin in alcohol solvent, this study addressed the evaluation of structure and corresponding quercetin properties on the interaction between the compound in alcohol solvent. Especially we use methanol using molecular dynamic simulation. So let's dive in. Steps involving in the molecular dynamic simulation was initialized by assigning the initial coordinates and then followed by the structure minimization to find out the lowest possible energy conformation. And then the solvation was done if explicit solvent was used, and also the initial velocities assignment was done after this solvation. Upon heating dynamics, we use temperature of 300 Kelvin as optimum temperature for the biological living things to work on their mechanism. And then after that, the equilibrium dynamics such as the control of temperature and structure was set, and if the temperature and structure was okay, the production dynamics such as NVE, NVT, NPT ensemble was used. And then in the last, the analysis of trajectory and calculation of macroscopic values was done at the end of the molecular dynamics simulation.
Real molecules vibrate all the time. Single bonds rotate, they tumble, twist, and turn in their space. Here, you can see a quercetin molecule is moving around by heating from 0 to a temperature of 300 Kelvin without solvation. It is actually much slower than the actual, but it gives us an idea of how molecules move and wiggle around. A model of quercetin dissolved in methanol solvent is displayed here. The method chosen was box with box size of 25 angstrom, big enough for the size of quercetin compound, and the solvent model is MEOH box, which stands for methanol. These can be seen from the abstract-looking structures around the molecule. The reason why we use methanol is because according to Shaves et al., 2020, generally methanol is one of the organic solvents used to extract flavonoids such as quercetin itself from plant matrices. And as you can see, the molecule's movement is faster than before since solvent is added to it. We'll see more of the comparison on the next slides. Here are two graphs showing the effect of quercetin RMSD upon trajectory frame number. RMSD is an abbreviation of root mean square deviation, where it has threshold of between 0.5 and 3 in this case. The first graph shows the RMSD values of unsolvated quercetin, where it specifies stability between frame number 550 and 800. And after solvation, the RMSD value is said to be fairly stable along frame number of 200 to 950. Both graphs shows RMSD of below 2.0 angstrom, hence considered to have a good RMSD value. Now here, the two graphs show quercetin effect of temperature upon trajectory frame number. The temperature was increased from 0 to 300 Kelvin, where it was constant at the first 500 frame numbers, then became inconstant after frame numbers of 500 till the end. The highest temperature reached was 400 K at frame number around 650 before solvation, and after solvation, the highest temperature reached was 450 Kelvin, which is higher, at frame number around 580. As for simulation with regards to potential energy, the potential energy in this situation is defined as energy in which particles in simulation interact. As you can see from both graphs, the energy shows inconsistency from the beginning of trajectory frame until the end. However, between frame numbers 550 to 1000, the energy gets a little more constant between 230 and 280 joules. And that's all from us. Thank you to Professor Mokhammad Fajar Pradipta, S.Si M.Eng, as our molecular simulation course lecturer, and to everyone who have listened till the end. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.